Hello students, welcome to my class. I'm Keerti. Hope you all are doing fine. Today we are going to have a very short session about the topic fermentation and antibiotics, right? So this topic comes under the chapter name Microbes and Human Welfare, right? Earlier we have a discussion how microbes are affecting our body, how they are infecting us. It means we have talked about the disease causing microbe, right? So there were many microbes like for example bacteria, fungus, some viruses that were adversely affecting our body. But we also have good bacteria, right? We already know these microbes are present everywhere, right? Even inside our body, the gut is having a microbiome of all the bacteria. Okay, our stomach is also having bacteria, right? So these these are the microbes which are actually helping the human beings to survive in a better way, right? So in this chapter we are going to study how these microbes are helping us, right? So let's start the topic. See, this fermentation and antibiotics both play a vital contribution. Both play a vital role in the human welfare, right? Fermentation helps to produce food and beverages, while antibiotics fight disease causing microbe, right? You already know whenever you are having some kind of disturbance inside in your body. Okay. And it is not cured on your own. You go, you see a doctor, and doctor prescribes some kind of medicines, right? And mostly you have heard this term antibiotic. Okay. Each and every doctor gives you some kind of antibiotics to get rid of that particular situation, right? So we will first study what is fermentation and then we will jump to our antibiotics. These are very short topics. Okay, you will be able to grasp this topic very easily. Fermentation is what? Fermentation is a metabolic process in which organisms convert carbohydrate such as sugars into alcohols or acid. These, micro- these microorganisms may be a bacteria, may be a fungi, right? Depending upon the type of raw material they are using, the type of organism what which is involved, right? So they will either produce byproduct like alcohol or acids, right? So these fermentation is usually of two types, right? The alcoholic, why? Because our end product is alcohol and carbon dioxide, for example, in case of wine and beer, right? And lactic acid formation, for example, curd and yogurt, right? You are well aware of the fact that in our home, we are eating many of fermented food, right? For example, the curd. You already know if you are, going to add some inoculum, it means the curd, a small amount of curd into a warm milk, right? And you will leave it overnight. What will happen? The curd will be formed, right? So the small amount of curd, when it is added, what is happening? Because of the suitable temperature, because of the warmness of the milk, right? This lactic acid bacteria that are causing this conversion, they will convert milk into curd. And what will happen? It will increase the nutritional efficiency. How? By increasing the vitamin B12 content, right? So you have seen that because of this fermentation, a new additional vitamin is added, which is helpful to us, right? So this picture shows that what type of organism is causing, is used, right? What type of fermentation products we are getting if we are going to use these particular type of organisms, right? For example, you are aware of the fact that this Saccharomyces cerevisiae, it is known as brewer's yeast, right? It is used to produce various beverages that contain alcohol, right? For example, wine and beer. The lactobacillus, the our curd bacteria, right? So it will produce lactic acid. Okay. So these are actually in a way enhancing the final product, right? So these fermented drinks, you can say why these alcohol, these, fermented drinks when taken in a very short amount, this act as medicines because the, alcohol content of this beer is very low, right? So what fermentation is yielding us? This is yielding us a product which have, you know, have enhanced nutritional quality, right? For example, milk to curd conversion, right? It is adding vitamin B1 to vitamin B12, right? And this fermentation is being done at industrial level also. When we are at home, we use this fermentation for many purposes. For example, you have eaten the idli, right? The dosa, which you already know. We keep the batter of that idli and dosa is actually fermented batter, right? We prepare the batter one day before, we leave it, it get fermented, and then we use it in the next day, right? So those are home products that we are having. But some of the products require a large scale production, right? For that purpose, what is happening? This fermentation is being done at an industrial level, and that are called fermenters. Fermenters are, for example, the vessel in which this fermentation is going to carry on. Because see, this production is at very large level, not for 1 or 2% or say 10% or 15%. For thousands and thousands of person, this process is used to produce a particular product, and that product maybe, for example, if we are taking into account the beverages, the wine, beer, whiskey, right? So this ethanol formation is taking place because of the yeast that we are using, right? For example, if our raw product is different, our end product will also be different. The type of raw material what you are using, the product will be dependent upon that. If you are using, for example, the fruit juices, you will get wine. Of course, there will be an additional process, for example, distillation. Okay, this is being done to, obtain a different product. So some of the beverages are produced without distillation. They are used in that form only, and some of the products are used after distillation. Okay, distillation is what? Distillation is heating of that compound to evaporate the volatile uh, compound from that solution. So you are having a solution, you will heat that solution, and the most volatile compounds in that solution will evaporate. So the remaining product will be taken out. It will, we will do all the processing and recover that product, right? So why this fermentation is required? This fermentation improves the food preservation, right? It also enhances the taste, the texture, and in some cases, it is also adding some kind of nutritional value, as we have seen in the case of curd, right? So there is always enhancement. Okay, so these this process of fermentation, we can use it to improve the nutritional quality of a particular product, and then we can consume it, right? And when we talk about antibiotics, you are already aware with the term antibiotics. You have, heard it. Maybe you don't know it, but you have heard it many a times, right? Anti means opposite, and bio means living, right? It means something that is acting opposite, or acting against to the living organism. So these antibiotics are actually derived from the microorganism and are used to kill or alter the nature of the other organism, right? So they will be, obtained from the microbes, or, and what will they do? They can kill or inhibit other harmful microbes. Actually, they are obtained from the microbes and will kill other microbes, right? This discovery of this antibiotic first happened when a scientist known as Alexander Fleming discovered penicillin, right? From a fungus Penicillium notatum. And it was a, chance by chance discovery. He accidentally discovered this uh, antibiotic, right? Later on, Chain and Flore, they discovered it. They have done, experiments. They have discovered more of the aspect of this antibiotic, right? So why this antibiotic is needed? Because see, our body try to eliminate any harmful pathogen that is coming out from outside from our body, right? From the environment. But in some cases, it cannot overcome it, right? So in that case, we are required to have some kind of product which are taken from outside to inside to create the balance, or to, you know remove that imbalance. Okay, so these antibiotics in some of the, rare diseases, for example in plague, diphtheria, leprosy, whooping cough, these antibiotics were very helpful, right? For example, earlier when a population is suffering from plague, all the human beings will die. Nobody can be saved. But after the advent of antibiotics, these people can be saved by administering antibiotics to them, right? That is why these antibiotics are very important to us. And there are various type of antibiotics. For example, streptomycin, tetracycline, erythromycin. They are either derived from the bacteria or fungi, right? Because you already know viral infections cannot be cured by administration of antibiotics, right? That is why, even if we have developed this much, the science has developed to this much, we are not able to find the cure of the normal, uh, cold and cough, because it is caused by rhinovirus, right? The medicines that we take during this cold and cough, right? These are just to depress the allergic reaction that our body is having. It is not actually depressing the viral multiplication, or it has nothing to do with this viral infection, right? That is why the cold and cough is very common. Okay, so since these antibiotics are very important, and now the population has grown very much, we need a mass production of antibiotics. Okay, that is why the industry, the pharmaceutical industry is there to play the role. They are cultivating antibiotics in a very, very, very large amount, so that it can be transported from the site of production to wherever or whichever place where it is required, right? So this figure shows a simple pathway how antibiotics are produced, though it is actually consists of various number of steps depending upon the type of inoculum you are using, right? How the final product is recovered, how the purification of final product is there. So that is something beyond our discussion level, but we will see how this is actually traveling, how it is being done in a very, very simple way, right? Suppose the lyophilized culture, it means the dry form, ice dry form is used as an inoculum. Inoculum is nothing when the first amount of that particular type of, say, suppose if you are taking the bacteria which are going to produce the particular type of uh, protein, the antibiotics that they are going to produce, so that inoculum consists of a very small amount of that bacteria. We will add it to the medium. Medium means what? The culture medium in which they are going to grow, they are going to eat the food, and they are going to multiply, right? Okay, it is added to the pre-fermenter. Now being transferred to the fermenter, right? Various nutrients will be added to this fermenter to support the growth of that particular microorganism, right? A kind of filter is used. Here it is written mycelium. Okay, and then it will be transferred to the cooling track. Now the product, suppose if it is the extracellular protein, if a bacteria is synthesizing some kind of extracellular protein, it means it is being secreted outside the cell. So if this is the medium, this medium will have your desired protein along with the bacterial cells, right? So this protein must be extracted from this medium. So there will be steps that are involved in purification and recovery, right? So this is just an overview to have you some kind of idea that how these, in at industrial level, how this is happening. And these are taking place, at a very large scale, right? So of course, the advent of antibiotics have revolutionized the medicine by controlling infectious diseases and saving millions of lives. Earlier if a person is infected, right? So it may happen that this is a viral infection, the other healthy person will may get infected, and because we didn't have any cure at that time, so the person will die, and this disease may spread, so whole the population will die, right? For example, in case of plague. But now we have advanced so much. We have developed so many techniques. We have developed so many antibiotics for a for each, depending upon the type of disease you are suffering with. So actually, many of the lives have been saved because of this antibiotics, right? Nowadays, we know that genetic engineering has come to in place, right? So what they are doing, they are actually enhancing the antibiotic yield. How? By designing various kind of experiment where we are going to increase the yield of the antibiotic. And in some cases, producing resistant free drugs. Right. So by designing such kind of experiments where we are going to alter the gene of a particular organism, say in case of bacteria, suppose if a bacteria is producing four proteins after one division, right? Four proteins. But we have done some kind of genetic level change, right? Now, after one division, this bacteria is actually able to produce eight protein. So what we have done, we have doubled the protein production, right? So this is how genetic engineering plays a role. Okay. So overall, what we can say, these two products that we are using in our day-to-day life, this, for example, eating the fermented food, right? And the antibiotics that are now there to save us, these are actually dual role of microbes. First one is as a food producer, and second one is as a lifesaver in medicine, right? So hope this is clear to you. Now you can say that microbes are not only, harming us. Actually, there are very microbes that are actually, you know, helpful to us, right? Hope I'm clear. Okay. So this is a very short topic which we have covered. I request you to go through these slides and also through your NCRT. And if you have any doubt, you can contact me through my Urban Pro profile. Right? Till then, keep learning and take care. Thank you.